Hi everyone, welcome to my new webinar. Today our guest is Professor David Hibbett, who is a mycologist at Clark University. His research focuses on fungal evolution and diversity, especially how different fungi or different groups of fungi are related to one another. He also leads a research lab that uses DNA-based tools to trace the evolutionary history of Agaricomycetes, which are the mushroom-forming fungi. Thank you, Professor Hibbett, for being part of this interview. Your insights are very valuable to us.
Thank you, Ida. I'm happy to be here.
Before asking any specific questions about your research, I'm curious about your story. How did you first get interested in studying fungi?
Um, well, I mean, it was very serendipitous, right? I went to college a very long time ago in the 1980s, and even before that, as a child, my parents sent me to Mass Audubon Society camps, and those were nature camps. And I grew up next to a body of water, a pond in Arlington, Massachusetts, and so I was always in the pond and around the pond and in the woods. And so I think I've been a nature freak since very early childhood. I was always interested in bugs and plants and fish and stuff like that. And in college, I was a botany major. You could be a botany major back then. And I took classes in plants, vascular plants, bryophytes, also algae and fungi, of course. And so I had this very sort of general botany training. But I didn't figure out I was going to be a mycologist until I was actually in graduate school. So I had gone to grad school intending to study plants, vascular plants. And it's a long story, but basically the first project I was working on just didn't work out. And so I needed a new project, and my PI, my supervisor, a guy named Ritas Vilgeles, was and is a mycologist. And so I thought, well, I need a new project since the first one isn't working. And I'm working in the lab of a mycologist, and I've always been interested in, well, mushrooms and everything botanical. So that was it. So I switched quite late in my training to a mycology program, but it was very nonlinear, very serendipitous. I never planned to be a mycologist.
I know that your research kind of helped bring molecular phylogenetics into the center of mycology. So, for my readers who are not specialists, how would you explain what molecular phylogenetics is and why it's so important for studying fungi?
Yeah, I would rephrase your
Yeah.
premise a little bit. It's not so much that my work helped bring molecular phylogenetics into mycology. It's that I was very fortunate to be working in grad school at a time
Yeah.
when molecular phylogenetics was just blowing up.
Mhm.
And I'd always been interested in systematics, and I was interested in using whatever tools were appropriate. So whether it's chemotaxonomy or morphology, ultrastructure, whatever. Um, and molecular methods became available while I was in grad school. A molecular biology technique called the polymerase chain reaction, or PCR, was published while I was in grad school. And so it kind of fell into my lap. And so it wasn't anything intentional that I did to bring molecular phylogenetics into my college. It just sort of happened while I was working. Um, but it's incredibly important because the molecular phylogenetics is using DNA sequences, so gene sequences, to estimate evolutionary relationships between organisms. And all organisms have DNA, and they all have certain genes that are shared across all of life, like the genes that encode ribosomes, the protein-making um, structures in the cell. And so you can use the sequences of genes like that to compare organisms that are morphologically really different, So you can make phylogenies with bacteria and sunflowers and blue whales and mushrooms in them using, ribosomal RNA gene sequences, but you're not going to find any morphological characters that will, well, actually, that's not entirely true. You're not going to find many that are going to help you with those problems. So it's really important because molecular characters, for the first time, really let us start to get an accurate, detailed picture of the fungal tree of life, the relationships among different groups, and to actually begin to use statistics to estimate our confidence in the relationships. So that's why molecular phylogenetics was and is so important for mycology.
Mhm. And has understanding fungal evolutionary history changed how you interpret the roles fungi play in ecosystems in any way?
Yeah, absolutely. I mean, I think when you study evolution, as I do, especially over long time scales, you come to understand that lineages aren't static. They change through time. And so when I see an organism out in nature doing something, I think about how that lifestyle evolved, and what it's doing at a sort of a like if it's a mycorrhizal fungus, what is it doing in order to be able to satisfy its carbon requirements? And how is that different from, say, uh, a closely related fungus that might decay wood or leaf litter or something like that? So absolutely, I think about those kinds of transitions. And when I'm out in the woods walking around looking at mushrooms, I'm thinking about their relationship. So I'm not just grouping them morphologically, I'm thinking about how they're related to one another. And so I see them not just as these sort of static entities, but as the very tips of the tree of life, which is continually growing and
Mhm.
has been growing for hundreds of millions of years. So I think an evolutionary perspective has a profound impact on how one views biodiversity in general.
I'm also curious about what you are currently working on in your research.
Yeah, so as you mentioned in the intro, I have a lab at Clark University. Right now, I have two PhD students and a postdoc in the lab, and a group of undergraduates, really terrific students, and they're working on a bunch of different projects. So some people are working on oyster mushrooms.
Mhm.
Some of my students are interested in the golden oyster mushroom, which is this invasive species that's spreading across North America. One of my PhD students studies nematode trapping behavior in oyster mushrooms and their relatives. Um, a bunch of us are focused on a genus called Lentinus, which includes Lentinus tigrinus, the tiger sawgill mushroom, and that's really interesting because it's dimorphic. It has a gill form and a puffball-like form that's called the secotioid form. And so we're really interested in the evolution of that morphological innovation and the genetics. So we're using a lot of population genomics and genome-wide association studies to try to get a handle on the molecular basis of this transformation. And I gave a talk on that for NAMA that if anybody's interested and they can find online.
Looking back, what kind of has been the most surprising or conceptually important discovery you have made while studying fungal communities?
Yeah, that's an interesting question. I don't know that I've made any conceptually important discoveries. I really feel like I've been, my work has been really just, describing nature and
Mhm.
inferring relationships. So I don't know if those count as conceptual discoveries. Um, I think one of the most interesting ideas that I've ever had, to the extent that I've had interesting ideas, some many years ago now, over 20 years ago, working with a Swedish grad student named Henrik Nilsson and some terrific computer science undergrads at Clark, we developed this tool for doing what we called automated phylogenetic taxonomy. So we built this little program that would go to GenBank every week, the NIH-funded repository of molecular data, and would grab ribosomal RNA gene sequences of fungi that Agaricomycetes, the mushroom-forming fungi, and add those new sequences to this sort of ever-growing alignment of sequences and phylogenetic tree. And then it would parse the tree. It would divide it up into branches using an algorithm. Um, and then it would write these little web pages and update these web pages about, the contents of various branches of the tree. And it was very rudimentary. It was very hacked together. It was a student project, not to disparage the students. Um, but I mean, I think it was a really exciting idea. The
Yeah.
idea was that if taken to its ultimate potential, what you would have is an ever-growing online tree of life, comprehensive, all the data,
Mhm.
um, and a system not just for building this giant tree, but for parsing it into branches and saying what's in the branches. Um, and it annoys me that 20 years later, more than 20 years, we published this paper in 2005. We still don't have that. So we've got Google Earth and so on, but we don't have, the tree of life is not online. there are trees of life online, but none of them have realized, the vision that we had back then, which is that you'd have something to be growing in real time, continuously updated. And of course, the phylogeny, the tree,
Mhm.
is a great tool for organizing information. So you could use the tree as a structure for pulling together information from whatever sources, GenBank, genomes, iNaturalist observations, whatever. So I think that was maybe my most interesting idea that
Mhm.
I've had. And unfortunately, it hasn't really been picked up in a big way.
And looking ahead, what do you think is the most important question that the next generation of mycologists should be tackling?
For me personally, I think one of the big questions is how do fungal forms evolve? We have there's this field called EvoDevo, evolutionary developmental biology,
Mhm.
very advanced in plants and animals, but not so much in fungi. So, mushrooms make all these incredibly beautiful, diverse, complex forms, and we really don't know how they evolve. We don't know how the developmental programs work and how they shift through evolution to give you a transformation between, say, like, a coral fungus and a gill mushroom.
Mhm.
How do those little hyphae know what to do in order to make a mushroom? I mean, mushrooms are just incredible things. They're basically an emergent property of the apical growth of lots of hyphae. And how do the hyphae know?
Mhm.
I think we really don't have a clue there.
Yeah.
Um, we know a lot about how fruiting body initiation starts, and we have some mutants that tell us something about certain features. But we don't have any kind of holistic picture of mushroom developmental evolution. So that I think is a big
Mhm.
one. But there are many others, many other sort of grand challenges of mycology.
So the last question is specifically for my audience. Many of my readers are not are enthusiasts, early learners, but not professional scientists. What advice would you give to these people who want to learn more about mycology or maybe even do research one day?
Yeah. Follow your blog.
Yes.
Um, no, seriously, there's an incredible amount of online resources that are
Mhm.
available, so you should check that out. It's a fantastic time to be interested in fungi, um, whether you want to do mycology as a career or if it's just going to be an avocation. It's a fantastic hobby. Um, I think the people who have it really good right now are people who are, mycophiles, fungal enthusiasts who aren't necessarily trying to make their living doing mycology because there's so many resources. There are so many clubs, regional clubs. The North American Mycological Association, NAMA, has a directory of clubs all over the country. For us here in Massachusetts, we have the Boston Mycological Club, which is a fantastic group. There's Pioneer Valley Mycological Association out in Amherst in the Happy Valley, and others around us. There's clubs in Rhode Island, there's clubs in Connecticut. And the clubs are fantastic. They have wonderful people involved who know a lot about fungi and really want to share their knowledge. So join a club, go out on forays, listen to lectures, talk to people. It's educational and it's social. So that's important. Um, there are also a bunch of good books now that are out. There are some really good mycology books. In recent years, there's sort of this new crop of books that have come along that are grounded in science, but written in a very engaging sort of way. So, like Entangled Life by Merlin Sheldrake is fantastic. I like Mycophilia by Eugenia Bone. Gordon Walker has a good book, Passport to Kingdom Fungi, and there are others. So there are some very good resources that are out there. Now, as far as doing research, that's harder, right? Because if you're talking about being a professional mycologist, um, there are opportunities, but they're limited. Like so, a lot of us end up in academia. Academia is a very stressed industry right now, so there aren't a lot of jobs. Um, a lot of us end up in government labs working for organizations like the USDA, and federal science right now is under a, full on attack. It's being downsized horribly, so it's not great. So it's kind of a tough time. Museums are another place where mycologists have found employment. Museums are also stressed. So it's challenging, but it's doable, and if somebody really has a passion for being a mycologist, then you only have one life, so go for it, I would say. But it's not an easy thing to make a living as a professional mycologist these days. But on the other hand, if you're a mycophile, the resources that are available now are just unbelievable. So great time to be a mycophile.
Thank you so much for taking the time to speak with me today. I really enjoyed learning about your experiences and your insights, and
Yeah,
I
my
believe all
pleasure.
this information will be very valuable for my audience as well.
Great. Thanks for inviting me. It was fun talking with you. And if you or members of your audience have questions for me, don't hesitate to get in touch.